Hello, everybody. I'm Winslow Hansen, assistant scientist here at the Carey Institute, and it's my pleasure to host Dr. Laurel Lynch of the University of Idaho, who's been here all week. It's been really fun. Thank you for coming. So I met Laurel a couple years ago when I went to a workshop at the University of Idaho, and we got along. We had some good science talk. We had some bike rides, and that's continued and been really fun. But one of the things that I've been most impressed with Laurel is she has this intense creativity and this analytical capacity to follow through and execute really solid and novel science at the intersections of ecosystem ecology and microbial dynamics. And in addition to the computer-based work that is so wonderful, I think Laurel might be the best field biologist I have ever met. And so if you're deep in the woods on a Halloween, and the ghosts and the ghouls are messing with your research, Laurel is the person you want by your side. So with that, I'll pass it over to Dr. Lynch.
All right, thank you, Winslow. So of course, I want to start by thanking everyone for being here today and for Winslow for being such a wonderful host. It has been so much fun being here at the Carey Institute. I've, of course, as an ecosystem ecologist, heard about this institute for many years, and it is such a pleasure to be here and to get to interact with some of the scientists and postdocs and researchers that are, really driving some very awesome research. So thank you for having me. So like I said, my background is in ecosystem ecology and biogeochemistry, and a lot of my work has kind of assessed how different disturbances influence ecosystem function, whether that's in Arctic and alpine environments, looking at shrub invasions, or how warming is impacting kind of rates of soil organic matter, turnover and microbial processing of carbon, to now working with Winslow and with Tara and a number of other colleagues in Western conifer forests, looking at how compounding stressors from drought and wildfire are kind of working together to push biological systems possibly beyond their adaptive capacity. Right, so can we predict when forests will recover from these compounding stressors and when they will fail, and is that linked to microbial drivers in the phyllosphere and in the rhizosphere? And as I became more kind of engaged in the scavenging work, I really realized that as an ecosystem ecologist, most of my training was focused on how plant litter inputs cycle through soil systems. And on one hand, that makes a lot of sense because carbon transfer from forests or above ground plant communities into the soil is this massive flux of carbon. So as a result, we've built ecosystem models that are well parameterized to kind of predict how rapidly that litter will turn over, which microorganisms might be driving or catalyzing some of that litter decomposition. And then we're getting a better handle on the proportion of that litter-derived carbon that might be used by microbial communities to kind of build biomass versus the proportion that might be respired back to the atmosphere as CO2. And plant litter inputs, I think, are interesting because they have such a broad kind of variability in their quality, but there's a big stoichiometric mismatch in terms of their carbon to nitrogen ratio kind of entering this soil system and then microbial nutrient demand. So we think that a lot of that extra carbon kind of gets kicked back to the atmosphere as CO2 through overflow metabolism, and it's kind of this leaky or inefficient carbon cycling system, which got me really excited about animal inputs. Right? So animal inputs are a much higher quality resource, but I think that they've been largely overlooked by the field of ecosystem ecology. Again, on one hand, this is a very small carbon flux relative to plant carbon inputs. So instead of 60 billion tons of carbon entering the terrestrial system annually, we're looking at only about 2 billion tons of inputs. It's also heavily skewed towards human biomass and livestock, right? So we're kind of losing a lot of our wild animal populations and biomass inputs. It's a really ephemeral resource, so we think that maybe it doesn't matter because it's patchy in its distribution and it gets recycled very quickly. So are there any effects of these inputs on larger scale ecosystem dynamics like plant growth or microbial metabolism? But I would argue that it is a very unique resource as well. So it has a lot more nutrient availability that more closely matches microbial demand, and so it might be processed more efficiently by the microbiome. It also brings together this whole guild of scavenger animals, from vertebrate mammals and avian communities to invertebrates and microbiomes, both in the gut of the animal when the animal died, as well as all of the soil microbiome effects as they're starting to colonize and break down these carcass inputs. So it's this really cool arena for complex food web dynamics and metabolisms to all manifest at a single point in time and space. So these were the ideas that really helped us frame out kind of our fundamental research question, which was how does substrate quality impact things like microbial metabolism and microbial carbon use efficiency, and does that drive factors like soil organic matter formation in different ways? So on the one hand, in our litter inputs, we might think of this as a more kind of inefficient carbon cycling system, where this really high carbon to nitrogen resource is entering the soil environment. A lot of that extra carbon is getting respired back to the atmosphere as CO2. If the stoichiometric mismatch is really high, it might even force these microbial communities to invest in more enzyme production that actually turns over existing soil carbon stocks through kind of this priming loop. And so we might have a less efficient system in terms of forming stable soil carbon pools. And in contrast, on the right-hand side, we might expect that these higher quality carcass inputs are going to be used by microbial communities with relatively high carbon use efficiencies, which means that those animal inputs get taken into the microbial loop. A lot of that carbon and nitrogen is being used to build biomass and for growth, and less of that carbon is being released back to the atmosphere as CO2. We also know now that a lot of the microbial byproducts of metabolism, whether those are enzymes, metabolites, or dead necromass, are kind of sticky chemically and are more likely to interact with minerals and contribute to formation of these more stable soil carbon stocks. So this was really kind of the way that we set up initially these dynamics between carcass inputs and ecosystem function. So you might be wondering why we decided to travel all the way to Tasmania to test some of these questions. And there are several different reasons. So this is a map looking at total above ground biomass, and the lighter the color, the more kind of a hotspot of carbon storage an ecosystem is. And so obviously a lot of the tropics kind of pop out as being areas of high carbon storage. And then Tasmania, way down here at the bottom of Australia, is also a hotspot of carbon storage in these really old, beautiful eucalyptus forests. So not only do they have high carbon stocks, but because the island is relatively small, there are some kind of similarities across the island in terms of soil parent material and climate gradients. So if you're trying to set up a transect study, it's a good place to work because there's lower kind of environmental variability in the background that might be obscuring some of the results that we're trying to pick out. And similarly, there are a whole host of endemic animals. So if you're setting up these transect experiments, if you're a population and a community ecologist, it's nice to basically have results that scale to the entire population. If you're working in Tasmania, because there are a lot of unique creatures there, the results that we have from this ecosystem study is broadly applicable across their entire range. So for this project, we are focusing on kind of leveraging this natural experiment that has been manifesting on the island of Tasmania for the past several decades. I, of course, knew about Carrie because of the ecosystem studies component, and I'm super excited that there are a ton of disease ecologists here as well, which has been a really fun way to kind of expand some of my research interests through interacting with this other field. So many of you probably already know more about DFTD than I do, but it is this transmissible cancer. They are very rare in nature, but DFTD has actually evolved 2 different times separately. In about 10 years, it has wiped out almost 70% of the devil population. It spreads through social interactions, so devils will also often fight with each other for resources, and when they bite each other in the face, DFTD spreads through the saliva. It's invariably lethal, so as soon as a devil is infected, they have about 6 months to live before they die. So as a result, we're able to kind of leverage this natural experiment to ask some of these fundamental questions, like what happens when you knock out an apex scavenger or predator from an island ecosystem. So one of our collaborators discovered DFTD in the early 1990s in the very far eastern part of Tasmania, shown here kind of in this dark red color. And DFTD has slowly spread across the entire island, knocking down populations as it goes. So this has created a natural gradient in devil population densities, which allows us to link how removing this competitor from the food web reorganizes trophic cascades and maybe impacts things like soil biogeochemical cycling. And another reason I got really excited about this system is that devils are fairly unique in terms of their scavenging abilities. So this is video that we took from one of the conservation reserves on the island. And not only can devils eat just an inordinate amount of carcass biomass in a single sitting, but they have a really strong bite force. And that allows them to break bone material apart, and they're able to ingest that bone material. So I think it's only devils and vultures that are able to perform osteophagy in the environment. And bone material is also pretty interesting because it is this rich reservoir of calcium, which is commonly a limiting nutrient, but very essential for plant growth. And for me, I was most excited about the fact that it is used to regulate stomatal closure. So if a plant is facing a drought, for example, if it has enough calcium, it's able to really strictly regulate stomatal opening and closure, helping it kind of regulate the amount of carbon fixation versus water loss. So by having calcium available in the environment through bone scavenging, you might be relieving a constraint on plant adaptability to variable environment. Bone also weathers very slowly naturally. So a large skeleton might take decades to slowly break down, and that calcium is being liberated kind of in very small amounts and very slowly. In contrast, if a devil comes around and it eats this carcass, it can excrete that bone-derived material across the landscape, and that calcium is much more bioavailable, right? So you're taking a process that would take decades to occur, and you're speeding it up, so calcium is returned in the order of hours to weeks. So for this project, the ecosystem team has really built upon decades of research from 2 of our main collaborators. Menna Jones is a professor at the University of Tasmania, and like I mentioned, she is the one who discovered DFTD initially and has spent much of her career looking at what happens from a community perspective as devil populations decline. And she works very closely with Andrew Storfer, who is a professor at Washington State University, which is only 8 miles away from where Tara and I work at the University of Idaho. And Andrew and Menna have, again, kind of looked at coevolution of the tumor and the host over time. They've looked at community dynamics, but they hadn't added in an ecosystem component. So that was really our task. So if we know that devil declines induce these trophic cascades, do they extend below ground, right? So by changing carcass availability on the landscape, are we altering inputs of nutrients that influence microbial ecology and invertebrate scavenging behavior? How does that loop influence soil organic matter formation and persistence? And can that scale up to influence things like vegetation productivity? Okay, so here is our big team of researchers, led by myself, and collaborating very closely with Mike Strickland, who was Jane's former postdoc advisor and a wonderful microbial ecologist. The above ground team was led by Dr. Tara Hudeberg, who is here as well. She is a forest carbon scientist and modeler, ecosystem modeler. And we had a team of wonderful PhD students and postdocs, but I will especially mention Tori Stevenson, who is my PhD student and has done a lot of the research that I'll be talking about today. And then on the other side, we have our team of population and community ecologists. So again, that was led by Menna, who has tons of experience working in these systems, and with Andrew and Dave Crowder, who is an invertebrate specialist in their postdoc, Savannah Bartel. So with this work, we had two primary research questions. The first is, are there localized effects of carrion input and scavenging on soil biogeochemical cycles? And the second is whether those effects can scale up to influence things like ecosystem function. So I'll start at the smaller scale and first introduce you to our cast of characters. So of course, on the very top, we have our osteophagous devil, and then we have a host of mesoscavengers and predators on the island, which include the quoll, the possum, and unfortunately the feral cat. So in areas where devil populations decline, feral cat populations explode, and there are pretty devastating effects on the native marsupial populations. From the invertebrate perspective, there were a number of guilds that we looked at, but the ones that were most important for our study ended up being carrion beetles and blowflies. And of course, if you're going to do a carrion decomposition experiment, you have to have some carcasses. And so the patty melon became our experimental unit. They are a very small wallaby. I like to think of them as the white-tailed deer of Tasmania, which made me feel better about buying them from game hunters for $1 a patty melon. They are everywhere, and they are eaten by everybody. So for this localized effects of carrion scavenging on the landscape, we set up 4 primary experimental sites that span the devil density gradient. So in the far east, we have Blue Tier, where devil populations are about 15% of their original carrying capacity. DFT first infected these populations in the early 1990s, and so it's been about 20 or 30 years. The devil populations have been very low. Then we have a central site where devil populations are about 30% of carrying capacity, and a far western site where DFTD was just beginning to infect the population. It's in kind of the middle of its rapid decline right now, but when we were doing this work, there were only a few devils that had been infected. And then we also established sites on Bruny Island, which is disconnected from the mainland of Tasmania, and there have never been devils there. So this was kind of our background control. What do soil biogeochemical processes and microbial communities look like in the absence of devil activity? So at each of these sites, we set up a replicated experimental design where we had 12 patty melon carcasses. Half of them were staked into the ground and open to the entire scavenger guild, including devils. And the other half were put in these walled mesh cages, and the cage size was small enough to exclude scavenging by adult devils. We found out not juvenile devils, and I'll talk about that in a minute. So these cages were designed to allow scavenging by all of the community except for devils. So at each of these patty melon carcasses, we took soil samples from underneath the carcass and from 1 meter adjacent, again, as kind of that background control, at 0 days, 5, 10, 15, and 30 days of carcass deployment. We also had camera traps that were set up looking at each of the carcasses that are triggered when something as small as a blowfly whizzes past, as well as the native black rat populations, and then is, of course, very able to capture scavenging by the mesoscavenger community and by devils where they are present. And we also set up pitfall and fly traps so we can kind of sample the density and composition of invertebrate scavengers that were visiting the carcasses as well. So we had some really fun camera trap photos of devils in active scavenging mode. They are pretty amazing. When they find a carcass, if they are hungry, they can eat the entire patty melon overnight. So absolutely house it. On the far right side is a juvenile devil that is trying to eat an absolutely massive patty melon. And so we found that there were some really interesting disease-induced changes in demography that we weren't expecting, but ended up being pretty important in explaining some of our results. So Tacone was that central site where we expected devil population densities to be fairly intermediate. And what we found is that adult population densities were pretty low because of DFTD, but there were tons of juvenile devils everywhere. So what had happened, and we used unbaited camera surveys to look at the population densities of all of the animals at the 4 different sites, is typically a female devil will have her first litter at about 2 years of age, and then they usually die around 6 or 7 years. In areas where DFTD was really knocking down populations, females were having their first litter at about 1 year old, and they were dying by about 3 years old. So as a result, instead of having kind of this big age structure that was well spread across the population, there are very few adults and tons of little babies, which again kind of messed up our winter cage experiment because it could just slip right through. So the winter ended up being what happens when you exclude adult devils, not all devils. And the other cool thing that might be expected is that these juvenile devils really serve the same role as a mesoscavenger. So they don't have the bite force and they aren't large enough to really consume a carcass as rapidly as an adult. They're also very furtive. So if they hear an adult, they are gone immediately. So they aren't super efficient scavengers. They're kind of looking up and being very vigilant. So inhabiting kind of this lower niche. So with all of that aside, what we found is first we're looking at carcass persistence. So on the y-axis is the percent of the carcass that was consumed. And then on the x-axis is the number of days of carcass deployment. And then it is faceted where you've got the open carcasses that were available to the entire scavenger community on the bottom panel and carcasses that were excluded or that excluded devils on the top panel. And what we see is that in areas that have higher devil densities, so again, these darker colors, carcass consumption is pretty fast, and it's particularly fast when you have devils that are present and able to access the carcass easily. So if we look at this 5-day mark at Salmon River and at West Tacone, between 75% and 100% of the carcass is consumed within that 5-day period. We do see a lot of carcass consumption by the end of the experiment in both of the sites, but the rates of carcass consumption are a little bit slower when you don't have devils in the environment. You also might notice that the gray bars, that's Mount Mangana, and at 30 days, we don't have full carcass consumption ever. And the reason is because there are no devils at Mount Mangana, and so there's always at least skeletal material that's left behind. So all of the soluble, fleshy material has been degraded for the most part at those sites, but the skeleton remains intact. So this really became a story of substitution, survival, and trickery, which is kind of a good trick-or-treat scenario for Halloween. But here on the left is a quoll at one of the carcasses, and typically quolls are nocturnal scavengers and predators. But in areas where there are high devil densities, they change their foraging behavior and they become diurnal. So they're coming, they find the carcass during the daytime. If they discover it, they will sit on the carcass and they will eat all day long until it gets dark, and then they'd scurry off. And typically a devil comes in after and will often finish up the job. As I mentioned, with the demography changes, we still saw a lot of carcass scavenging and activity, even at kind of these intermediate and lower double density areas, but a lot of substitution by smaller, more juvenile devils. And we had a number of our caged experiments that failed for so many different reasons, which was a good reason that we had an N of 6 at all of our sites, so we could ditch some of the ones that didn't work. But this is one example where a devil found a carcass, really wanted it, dug a hole, and laid on its back for the entire night and just ate the carcass from underneath the cage. She's like, all right, you get that one. Okay, so I've been told that I have to include a trigger warning for the next 2 slides. So if you don't like Halloween type imagery, then maybe avert your eyes for a moment. But I think this is super cool because I had never seen a carcass in active decomposition, and so I was very weirded out and thought it was super cool and wanted to share some of these images. So first, this is still a patty melon, but it's naked. So in the winter, we found that there was tons of activity of hide beetles, which eat the dermis. So they kind of specialize on basically removing the hide of the patty melon. We also had tons of larval activity from blowflies and from carrion beetles as well in the winter. In the summer, a lot more of the scavenging was being taken out by adult invertebrates, mostly carrion beetles. So in both the winter and the summer, but particularly in the summer, one of the other notable effects was just the amount of moisture that these carcasses deliver below ground. So we're not only looking at nutrient delivery, but here on the right are, on the top, were soil samples that were collected a meter away from the carcass, and then on the bottom were samples collected directly below the carcass. So not only are you kind of bringing nutrients into the system, but all of the soluble material is making the soil a lot wetter, and in dry periods, especially microbially productive. So kind of an added benefit of localized scavenging on soil productivity. Okay, one more kind of intense video. But I also wanted to show how amazing invertebrate scavengers are. So this is looking into the pouch of a patty melon that is just writhing with blowfly larvae. So again, I'll move on so you don't have to look at it anymore. But when you come to these carcasses, like, you can see them moving and you can hear the larvae just breaking down this carcass, right? So again, there's this huge localized input or subsidy of nutrients and soluble organics from the carcass directly below that is really influencing biogeochemical cycling at this local scale. So again, on the upper panel, we're looking at sites where devils were excluded, and in the lower panel, at sites where devils were able to forage. And we find that when devils are present in the landscape, because they're consuming the carcass so quickly, there are fewer nutrients that are leaching into the soil below them, right? They're just getting ingested, and all of those carcass-derived nutrients are getting spread across the landscape through their excretion. In contrast, when you have carcasses that are sitting on the landscape for a long period of time, as they go through scavenging by these mesoscavengers that are less effective scavengers than your apex scavenger, or through this invertebrate and microbial-dominated loop, you have tons of nutrients, including nitrogen and phosphorus, that are flowing below ground and subsidizing microbial activity. So you see basically the same patterns with phosphorus as well. For nitrogen, this is mostly driven by ammonium input. So from a microbial perspective, we do see pretty obvious responses below ground. So this is an upset plot, which is basically just looking at the number of taxa that are shared between treatments and those that are unique to a treatment. So this first bar is looking at the taxa that were present, regardless of whether they were found kind of in the forest soil or directly under the carcass. And we see that there's a lot of redundancy. But if we look at taxa that are present only in the carcass and not in the forest soil, we find that there are very few that are kind of driving this process. If you look at PCOA plots and Shannon diversity and a lot of other kind of microbial metrics, what we find is that there's this transition from kind of balanced microbial activity towards microbiomes that are increasingly dominated by these faster growing phyla, right? So organisms that are able to capitalize very quickly on these pulsed resources, bloom in abundance, and kind of drive the biogeochemical cycling of these carcass inputs. When we went back to sites in the summer that we had set out in the winter, we still found that there were legacy effects. So the microbiome had not recovered. It still looked different than control soils if a carcass had been present. So even though visually on the landscape, carcasses appear to be an ephemeral resource, when they are present and leaching a lot of those nutrients below ground, there are legacy effects that persist for at least a year. So for our follow-up work, we are designing a stable isotope labeling experiment to get a better handle on what is happening below ground. So we're going to take our control soils and our carcass impacted soils, and on top of that, we'll add in 13C and 15N enriched plant litter and putrescine, which is a byproduct of animal decomposition, and look at how those different products cycle through the microbial loop, how those influence microbial carbon use efficiency, and whether they're really driving changes in soil organic matter formation and persistence. Okay, so really quickly, I'm going to jump to kind of the next stage of investigation, which, okay, we're getting kind of a handle on very strong localized effects of carcasses on soil microbiomes and biogeochemical cycling. But can those scale up to influence these larger scale processes that most ecosystem ecologists and particularly modelers would be interested in? So we're moving from kind of below ground to above ground dynamics. If you haven't been in a eucalyptus forest before, they are beautiful, but the trees are very, very tall. And if you have to get light lit foliage from the top of the canopy, you have to be somewhat inventive, especially in Australia where you can't have shotguns and just shoot the leaves down. So we had a giant slingshot and beanie bags and tried to wrench leaves out of the canopies of these very tall trees that are also very bendy. But we managed and have some good foliar nitrogen data that we used to parameterize some of the ecosystem models. We also collected about 200 tree cores from trees that are spanning the double density gradient. So we're able to look at changes in annual NPP as well as kind of biomass changes that we are hoping in the next stage of this work to link to changes in double population densities. So one of the first questions was, okay, we see these very clear changes in carcass persistence where we have devils and where we have lost devils. We know that devils eat a lot of carcass inputs very quickly, and then they are going to poop someplace in the landscape. So does the scat, and often devils will create kind of these scat latrines where the scat is very concentrated locally. Can leaching of nutrients from the scat also be a source of nutrient availability for the forest? It's a little bit more dispersed than what is coming directly from a carcass. So we set out scat latrines also across the island. We saw similar changes in below ground microbial community composition, same sorts of things where the scat was selecting for faster growing phyla and kind of more rapid biogeochemical cycling. And also localized increases in nitrogen and phosphorus and also calcium. And Tori worked with Tara and our postdoc Justin to try to scale this up. So Tara is an expert in DayCent, which is the daily time step version of Century, which is a very important ecosystem model. And we basically use two different scat qualities, one from devils, which have a much lower carbon to nitrogen ratio, and one from pattymmelons. They're an herbivore, so they have a much higher carbon to nitrogen ratio. And then we looked at if we are adding this scat across the landscape, can it influence some of these larger scale pools, very important carbon pools like above ground net primary productivity, below ground net primary productivity, microbial biomass carbon, and soil carbon. And then we also use downgraded climate data to force 2 future climate scenarios, one with kind of constant conditions, keeping temperature and moisture regimes similar, and then another where we force kind of this future scenario with a warmer and wetter climate, which is predicted for a lot of the island. In these teal colors are what happens when you continue to have double scat inputs in the ecosystem. Purple is if you remove those double scat inputs and you have primarily scat inputs from herbivores and some of these mesoscavengers. And what we see for pretty much all of the modeling scenarios that we ran was that devils have a positive or at least a modifying effect on carbon storage across these pools. So when you have devils, above ground, below ground net primary productivity and microbial biomass carbon remain a little bit higher, and the loss of soil carbon is reduced. So in these future modeling scenarios in Tasmania, it doesn't look great for soil carbon dynamics in general. Like there will likely be a loss, whether it's ambient or warmer and wetter. But when you have devils present in the landscape, the magnitude of that loss is reduced a little. So we are planning to continue testing some of these dynamics empirically. Again, we collected these tree cores from across the island. We have 2 questions that are kind of associated with this project. One is that there are these really interesting kind of landscape forest patches in Tasmania where you can have dry eucalyptus forests that are directly adjacent to wet eucalyptus forests, and you can literally walk from a dry forest into a wet forest in about a minute. The canopy dominant trees are the same, so they're all dominated by Eucalyptus obliqua, and the slope, the aspect, kind of all the topographical features look pretty similar. The soil parent material is the same, but they have very different chemical environments and biogeochemical cycling environments. And so we're looking at what happens if you have kind of a different climate. Are trees that grew up in this wetter forest going to be less resilient to future drought, or do they have a bigger window in which to adapt, right? So if you're droughting this environment, do they have some of the plasticity or resiliency or background kind of carbon storage reserves to weather those drought events more effectively than these scrappy dry trees that are maybe living kind of at the edge of their phenotypic plasticity? So in addition to kind of looking at these future drought effects, we are also going to see if we can use calcium again as that biomarker of devil scavenging. So taking periods of annual tree growth, 5 years before the devil decline, during active devil decline, and 5 years post the devil crash, and looking at whether concentrations of calcium change in that woody biomass over time. That would be kind of the needle in the haystack finding, but would be really cool. And if we do find that, will help to kind of explain, again, some of these mechanisms of drought resilience. So we're also testing the effects of carcass additions and drought directly on plant physiology in the greenhouse. So with that, I am very happy to take any questions that you might have, and would love any feedback, especially from the disease and ecosystem ecologists. So thank you.
All right, so we have plenty of time for questions. I'm going to be summarizing the questions, so let's keep them short and tight, please, so I don't have to struggle with that. And Laurel, you can call whoever you want, and I'll just do the summary.
Perfect. Jane.
Super awesome, super great for Halloween. I have a lot of questions. I'll just start with one is, what do you think about kind of the anoxic conditions that'll happen underneath these for a while? And yeah, how is that, are you guys looking at kind of redox reactions in that system? And yeah, how's that happening?
Yeah,
So really quick, repeating the question. So under the carcasses, are you looking at anoxic conditions and redox reactions?
That was a very good summary. Yeah, that is a great question. So in previous studies that our group was not involved in, One of my favorites are kind of these like beaver decomposition studies where for the first like 5 to 10 days that the carcass is present on the soil, there are very strong anoxic conditions that drive changes, especially in nitrogen transformation, that are really key. It also changes pH. So there's kind of a lot of these state factors in terms of soil chemical cycles and biogeochemistry that are really important and are definitely impacted by anoxic conditions. So in our soils, we did have some redox profiles. We didn't see the same kind of redox effect, and I think the reason is our soils were so duffy, like this really light, fluffy kind of moss layer. So I think there's still enough oxygen that's kind of flowing through that system that's offsetting like a really strong anoxic impact. But I think if you were working in other systems, maybe not sort of these really old forests, you would see those effects, and those would be really, really critical to get a better understanding on. If I remember right, a lot of the studies that are using kind of these smaller carcasses, the anoxic effects are fairly transient. Like I said, maybe like the first 10 days or so, and then oxygen is slowly kind of reintroduced into the system. But if you have larger carcasses, like there was an elephant carcass decomposition study or whale carcasses in the ocean, like that is a dramatic and probably very long-lasting effect. Yeah, that's a great question. Other questions? Yeah.
So in many of these systems where there's a large-ish mammalian scavenger, there are also plenty of other scavengers, smaller mammalian ones that you referred to a little bit, but also avian.
Yeah.
And there can be potential for compensation. Sorry, to what extent, I don't know the Tasmanian system well enough to know if there are other scavengers, but to what extent do they compensate for the declines and losses of devils, and what impact might that have on your nutrient and plant responses to that nutrient change?
Okay, so there's lots of different scavengers, large and small, avian and mammalian, etc. How do they compensate for the loss of devil through the cancer in terms of the biogeochemistry that you're seeing?
Yeah. So in Tasmania, there are a number of avians. We didn't get a ton of them at our sites. We did have, there's old like forest ravens that are some of the dominant ones. They were pretty gruesome, I have to say, where there was like an avian hit on their carcasses. It looked like a crime scene, like blood everywhere and patches of fur everywhere. And the same, there are some hawks and kind of wedge-tailed eagles that are present. So we did have some scavenging by avians, but in this system, it was kind of a lower contribution. I think you have partial compensation by some of these mesoscavengers. So again, we saw a lot of quoll and feral cat activity, especially at the sites where devil densities were low. We also saw a lot of scavenging by native black rats. So that was something that in kind of periods where these larger scavengers were not actively foraging, they'd be covered in rats. So there's sort of like a temporal substitution. Again, I think devils are unique in terms of the biogeochemical effects because they're uniquely able to ingest like so much of the carcass in one sitting. And so they're removing the carcass at a very fast rate compared to what the mesoscavengers are able to do. And again, the mesoscavengers aren't able to break apart that bone material. So in terms of nutrient subsidies and kind of changes in the biogeochemical profile, I would say there's less like calcium mobilization, but there's so much more nitrogen and phosphorus that is flowing below the carcass in areas where devils are less present. So I think that the localized effects are more pronounced when you don't have very many devils, and then kind of these landscape level effects are more enhanced where you do have devils. Yeah, it was a really fun, just so much fun to be in that system and see all of these different animals that hop and, do crazy things that we didn't know about beforehand. Yeah.
So that actually kind of aligns with the
Okay.
online question, so maybe we'll take one of those. So Chris, Flatbush Gardener, asks, are you aware of analogous systems in North America? So this is a pretty unique kind of case study, really charismatic creatures. Are there other systems where you could extrapolate these kinds of dynamics to in North America?
Yeah, definitely. So I would say the kind of richest literature for these like carcass decomposition studies are Australia and Africa, where you've got kind of these megafauna present on the landscape. So not historically, I shouldn't say that, 70s and on, that has been kind of a primary arena for a lot of this work. One thing that I'm interested in Idaho right now, we have a big issue with chronic wasting disease for white-tailed deer. So kind of analogous to a DFTD type of system. Of course, deer are not high up on the food web, but in terms of carcass subsidy on the landscape, there's a lot of input right now. So I think that would be an interesting system. And then in terms of the same kind of scavenger arena, depending on where you are, wolves versus coyotes versus martens, and kind of all of the mesoscavengers that would come into play as well would be really interesting to look at. Unfortunately, across a lot of North America, we're doing a pretty good job at suppressing wolf populations, and so we're sort of we've already suppressed this apex carnivore and scavenger on the landscape, which was one of the reasons that we were super interested in working in Tasmania. So these devil populations are in decline because of a disease, not because of human conflict or land use change or habitat degradation. So you're kind of able to disentangle some of those climate disease effects from human pressures. Shannon.
Two questions. One is about the demographic changes.
Yeah.
Do you know if do they recover back to the prior kind of demographic life history when the cancer's not in the location for a while? They do?
Do you have two questions?
Well,
Or should I answer them?
that's part of the question.
Yeah.
And even in the temporary span where it's all juveniles,
Yeah.
you kind of alluded to the fact that they're just different types of carnivores. Do you see an intermediate effect when there's a lot of juveniles, or do you just see it, or do they just look like the rats and other things at that point?
Okay, so if the cancer goes away, is there recovery of the demographic structure of devil population? And then if you only have lots of those little juvenile devils running around, what are the effects of that? Is it intermediary, or do they look more like other scavengers?
Yeah, so I'll answer the second question first. I think based on our results, they fit squarely into a mesoscavenger guild. So they're not intermediate between an adult scavenger and like a cool. They're about on the same quality of scavenger as the cool feral cat and raven kind of guild is. So our colleagues are putting in an LTREB proposal right now to look at basically repeatability and predictability of these devil population declines. So in the site that was first infected, they were really worried about localized extirpation, like is there going to be extinction of the devils? And there hasn't been. There's been very, very slow and sort of modest recovery. So DFTD has a very low presence in that population right now, but we're still only about 30% of the original carrying capacity. I would say one thing that is really unique and kind of hopeful about Tasmania is they mobilized so quickly to try and maintain some amount of genetic diversity within these populations. So as they saw DFTD was kind of spreading across the island, there were these huge conservation efforts where they brought healthy adult devils into conservation reserves, and there are many generations now of captive devils. They also have this island reserve where they bring populations that are close to extinction. So there's wombats and kangaroo and devils and all of these other animals that they ship off to this island preserve. When DFTD is kind of suppressed in a local population, they will take these captive devils and try to reintroduce them back into the wild to, again, inject some genetic diversity there and try to ramp up population recovery rates. One of the issues right now is that they finally were looking like they're going to make it through DFTD. And now there's an avian flu that is going through, and it's knocking back devil populations again. So it's a very vulnerable species. Even when DFTD is kind of not present in the population, it doesn't seem like they're coming back to carrying capacity very quickly. And they're super vulnerable to the next devil pandemic. Yeah.
So just to follow that up with an online question from Adrian, or perhaps Adrian is a he, I'm not sure. So sorry, Adrian. They ask, what are the causes of the cancers? Is it environmental? Is it intrinsic? Do we know?
I am not the right person to ask. That would be a Mena and Andrew question, and they definitely know the answer. And maybe somebody here knows the answer about DFTD. But it is a transmissible cancer, again, which is rare. It's evolved two different types. So they had DFTD that was moving through the island at a pretty rapid rate, and they started seeing kind of localized reinfection. So the main DFTD had already moved through. They thought the populations were going to be fine. And then devils started popping up with facial tumors again. And when they looked at kind of the genealogy, the evolution of these tumors, they found that they had created themselves twice, essentially, which is, again, really unique. I don't know exactly what their origin is. That's a great question. Lauren.
Are there big differences, do you think, in efficiency in converting a carcass to devil versus blowfly, for example?
Biomass?
Yeah,
Okay.
the process of turning it from carcass into one of those two organisms in terms of efficiency.
So are there differences in efficiency between converting carcass biomass to scavenger biomass, whether it's devil or blowfly?
Yeah, that's a good question. I'm just thinking now of kind of the energy pyramid, and as you move up through kind of the trophic levels, that you're having less and less efficient carbon conversion into biomass. I don't really know. I will say that I'm trying to think, like, what is their excretion rate and their metabolism rate and sort of all of those things. That's a great question. One thing that I think is really interesting in terms of biomass impacts, I hadn't learned very much about the invertebrate scavenging food web, but when you have a carcass that hosts, that kind of density of larvae, they can pick that up in like storms later, where if there's a big kind of release of the adult blowflies, for instance, that get caught in kind of a wind funnel, you can see their bodies and their carcasses kind of following that wind path. And there have been studies that are looking at the frass or the necromass of those invertebrates getting distributed across the landscape in these kind of directed storm-driven events that have legacy effects on plant productivity. So I do think that's. There's also differences in their dispersal rates, right? So just in terms of the excretion loop, devils have a pretty big home range. Carrion beetles aren't going to be able to carry the carcass-derived biomass very far before they die or pupate. So there's differences in kind of the concentric circles of effect away from a carcass, depending on which scavenger guild is most active. So I would say the long-term dispersal is going to be highest for the devils, but kind of intermediate for a blowfly and pretty low for a carrion beetle. Yeah, it's a great question. Tara?
So this is a kind of Halloween question, but I'm curious to hear what the landscape of carcasses looks like, and whether in your experimental studies you noticed or reported any variation in the density of carcasses across your study sites, and how that might impact the effects of these scavenger impacts on ecosystem processes.
That would be such a great follow-up experiment. So we spent a lot of time walking through the forest, and sometimes you would find carcasses that were not our own, but it was pretty rare. I think the consumption rate is just so fast that it is true that they are a fairly ephemeral resource on the landscape. We did kind of unbaited camera trap photography that, of course, is just looking at the living population kind of moving through. On Tasmania, I think the biggest carcass creator would be humans and their cars. So when you're driving through these kind of windy roads, like we did a carcass count for, just to entertain ourselves because there was so much driving, and you're like at hundreds of carcasses just lining the roadways. I think we created 20 or 30 from patty melons just waiting until you were right next to them and leaping under your tires like over and over. So I would say like there would be really cool kind of landscape patterns that are very linked to human populations and roadways. We were always curious, like, is the grass like especially nice because all of these carcasses have delivered their nutrients and now the grass is nice and perfect and all of these herbivores are coming to eat them and then contribute more carrion, and it's this like terrible feedback loop. But yeah, I think it's hard by impression, and again from reading some of the literature, like most people are deploying carcasses because they are so hard to find walking through a forest. Yeah.
So we have time for one more question, and I'm hoping I can ask it. Is that okay? So you're going to be shocked that when I see landscape mosaics of dry and wet forest, I think fire. But it made me realize I don't know anything about the fire regime. So what are like some alternative explanations for that weird mosaic of dry and wet forest?
Yeah, so we don't know for sure that it isn't fire, but in all of the cores, the tree cores that we took, we didn't see a single like burn scar in any of them. Some of the dry forests, you could see some scarring kind of on the bark, and I think that was probably controlled burn just to kind of clear things out. So Tara and I have been talking about this a lot, and we don't really know. It's kind of a chicken and an egg sort of scenario, but in the wet forest, these trees grow really, really huge, and their canopy kind of closes out. And it seems like it's creating, like it is a much more humid environment. You have moss everywhere, you have terrestrial leeches everywhere, big tree ferns. And so we were like, okay, maybe this is kind of a positive feedback cycle where you get large enough and you close off the canopy and you're creating kind of this wetter system. And adjacent in these drier forests, there's actually more trees, but they're much, much smaller, and they're like crowding each other out. So really skinny, lots of them, but very skinny, and their canopy does not close. And you have these like saw grasses and you have ants everywhere, and like it could not be more different. So I don't know exactly what started that loop of like large trees creating a more humid local environment versus lots of little guys creating a more arid sort of feedback environment. Fire is what we kept coming back to as well, but doesn't really seem to be strong evidence that is the mechanism. So we're still trying to figure that one out.
Interesting.
It is cool if you look at carbon storage and above ground biomass, they end up washing out and being about the same. So it's huge amount of carbon stored in a few giant trees in these wet forests versus dispersed across many in the dry forests, and we see very clear changes in like soil nitrogen availability, microbial biomass, RDK selected taxa, genes for nitrogen cycling. Like they are very, very unique ecosystems below ground as well. So we're still trying to kind of dial that in a little.
Cool. So we'll stay tuned.
Stay tuned.
Great. Well, let's thank Dr. Lynch for a wonderful seminar.
Thank you, everyone.
And just as a quick reminder, happy hour at 4 for anybody that's interested in joining and chatting more with our speaker.